Welcome back. This is Intro to Physical Anthropology. I am your instructor, David Leitner, and today we're going to talk about the brain, covering a little bit about sort of how technology has changed the way anthropologists understand brain evolution, and also some of the basics about brain terminology and anatomy. So without further ado, let's get started. So, lots of advances in technology over the past several decades have really opened up new possibilities for studying the human brain, and this has affected physical anthropology as much as it has fields like neuroscience and neurology and that sort of thing. Brain imaging in particular has given us a deeper understanding of the organization, structure, and the function of the different areas of the brain. Few of the types of imaging technologies that are very common, there's fMRI, which uses changes in blood flow as a proxy for brain activity, so it sort of maps out where more blood flow is occurring versus less, and the idea being that if there's more blood going to the area, it's carrying more oxygen there, which means that area is using more oxygen, which means, of course, that there's more brain activity, more cell activity, so more brain activity. EEGs map out the electrical waves coming off your brain. They don't literally come off, but your brain is an electrochemical organ, and it does have an electric field that can be sort of detected with electroencephalograms. CT uses X-ray slices, two-dimensional X-rays, but sliced across a three-dimensional object to build up a model of the brain as if you could sort of slice the real thing at any point. And you can see and build up a 3D model of the inside of the brain, not just the outside. Finally, PET, positron emission tomography, uses a combination of blood flow and oxygen use and sugar metabolism as proxies for brain activity. So, now these technologies haven't just given us insights into living brain tissues, they actually have helped us get more information out of fossil crania as well. We've talked about the Tong child before. This is one of Raymond Dart's original Australopithecine finds here. And the Tong child is not just the face portion of the cranium, but also has the endocast of the brain. Remember, an endocast is basically the impression of the inside of the skull, and that is itself a reflection of the bumps and the grooves on the surface of the brain, as well as the distribution of different blood vessels and so forth. And it gives you a lot of information about what areas of the brain are developing or not developing. That is a really useful feature because it lets us actually peer inside these things. Now, it doesn't necessarily preserve a ton of the internal structure, but by getting a better picture, say, with the Tong child, we're lucky enough to have a real endocast. With other fossils, we can't, it would be likely we would destroy the fossil if we tried to get the endocast. So in those cases, a virtual endocast does the same job. So it gives us access to information that was very difficult to get before. Okay, so that's a bit about sort of the tools that we use to study brain anatomy, but what is brain anatomy? We'll cover some of the basics here. First thing I want to cover, though, is some terminology that we'll probably use quite a bit. This terminology is pretty consistent through the medical profession, so if you're studying for a nursing degree or you're planning on going into med school or anything like that, you'll probably recognize some of these terms. Okay. Superior and inferior. Superior is above, inferior is below. posterior and anterior. Posterior is behind, anterior is in front. Then within the brain, we've got a couple of others. Rostral is towards the front of the brain, and caudal is towards the back of the brain. And ventral versus dorsal. Ventral is in front of the spine, or the thorax, really. And dorsal is towards the back. Okay. The only difference between these terms is really whether you're oriented to the body as a whole, so superior, inferior, or you're oriented towards a particular organ. So in the case of the brain, we see the brain and the spinal column as one continuous organ here. That's why it has a dorsal and ventral side that kind of shift a little bit midway there. Just because the orientation's a little different. All right. Here you can see a lot of those same terms, but so those are just general directions. Oftentimes when we're looking at images of brains, we're looking at sections, which is, you can think of a section as just a slice of the brain. So if we were to slice this brain in half along the green square here, the green pane, and split it open and look at it, we would be looking at a coronal section. Okay, so anything that sort of is in that plane is coronal. Anything that sort of goes from front to back like that is sagittal. Remember, we've talked about sagittal crests. Well, they're sagittal because they run across the top from front to back. And then finally, horizontal is just horizontal, which is nice. It's an easy one to remember. Finally, you've got the last direction, which is lateral, which is away from the center of the body, and medial, towards the center of the body. Okay? So, the brain itself is made up of two main hemispheres, okay? So it's like it's one big organ with two hemispheres that are connected in one small part deep down there. Both sides are more or less mirror images of each other, depending on what, at least physically. Functionally, there are a lot of things that are what we call lateralized. That means they occur more on one side rather than the other. This is sort of an adaptation to, this is a form of adaptation in brain evolution, as sometimes old functions, new functions piggyback on top of old ones, or they migrate to sort of create new ones. Now, apart from the hemispheres, there are three main sections of the brain, okay? You've got the brain stem, which includes the spinal cord and base of the brain, at the base of the brain, I should say. And it has the most basic functions of, like, just keeping you alive, right? Evolutionary terms, this set of structures in there are some of the oldest. When people talk about their lizard brain, they're sort of referring to this, so we can talk about it's not an exactly accurate way to think about it. It's just, this is the stuff you don't have to think about, like to keep your heart beating and, you know, regulate certain parts of your metabolism, that sort of thing. This is sort of the basic brain right here. The next section is the cerebellum. It's that little pink section underneath the big brain here. And it's cerebellum, which is, I believe, Latin for little brain. Again, scientific terms aren't that hard when you realize that they're just very literal. Okay? Cerebellum's important with balance, posture, and certain voluntary movements as well. So, very important part. Again, this is kind of the next oldest, the more recent, the next most recent structure in the brain. Okay. Finally, the biggest part of the human brain is the cerebrum. That is all of that other stuff you see there, okay? It is the vast majority of the brain's volume in most mammals. Actually, I think all mammals, really, you could say that for, but especially in Homo sapiens and in hominins in general, it tends to be much bigger. Um, this is where a lot of the higher functions come in, especially things like executive function or which includes planning ahead and um, keeping track of time, and a lot of social intelligence sort of is embedded in this area. A lot of our um, uh, a lot of our motor functions are, as well as our sensory input, are also sort of centered around the cerebrum. And this is the most recent sort of structure of the brain. Now, the cerebral cortex, or the cerebrum, as we just called it, has a number of different functional sections. That means that there are sections that perform different functions. These are sort of these sections actually can be subdivided down into other functions, but for the most part, everybody's got these functions in roughly the same area. The most interesting sort of the divisions here are the primary cortices, which primary meaning that they receive input. Feedback about motor control comes in the primary motor cortex, and sensory input is processed in the sensory cortex. Sorry, not processed, it's received in the sensory cortex. There are also other primary cortices in various places around the brain. Association cortices, on the other hand, do the analysis, okay? This is most of what the cerebrum is composed of. It takes all of that input and makes decisions, or sometimes it's instincts, sometimes decisions, but it is the sort of CPU, sort of, if we use the computer analogy for the brain, which is not a great analogy, it's not a perfect one, but to some degree it's sort of the CPU, sort of where stuff gets processed. And that includes things like the prefrontal cortex, where all of this executive function and social smarts come in, and the somatic association area, which is the premotor cortex, and the somatic sensory association area, which is sort of towards the back. You'll notice that the primary motor cortex, which is interestingly enough a map of each side of your body, on the left side of your brain is mapped the right side of your body, and on the right side of your brain is mapped the left side of your body. But it's a map of your body, literally, sort of, I think, starting from the head and going all the way to the toes. And connects to every muscle you've got down there, through the spinal column. And it should be no surprise then that we find the association area, the motor association area, right next to the primary motor cortex. Same's true with sensory association, sensory cortex, primary cortex. You can see the other sort of big major division here is at the very back in the occipital region is where both the visual cortex and then the visual association area are. These are always really close to one another because that's an efficient way to do that kind of processing. We'll talk about Broca's area and Wernicke's area in another video. They have to do with language production and comprehension. But you can see it's a pretty sort of complicated machine. Now, here's a coronal section, like I was talking about before, and most of it, you will notice, is this dense white stuff here. There are all these, did we talk about? I didn't mention the sulci and the gyri. So all of the wrinkles on the surface of the brain here, this is what they look like in cross section. They go rather deep in a lot of cases. The reason is to provide more surface area for this band of grayer sort of cells. They're gray on this. Uh, they're a little less. They're more of a pinky gray in real life, but they're more gray in this black and white image. More surface area means you have more of those cells going on. That's where the processing occurs. All of this white area it does seem to have some other functions, but one of the primary functions is to connect different areas of the brain, especially connecting down to the spinal column and the cerebellum and the brain stem there, as well as to other parts of the brain. This little area here and this one right here connect the two hemispheres together so that they can talk effectively to one another and coordinate information. Neurons. So when we break it down into the basic building blocks of the brain, the smallest unit is a cell, right? Just like the basic unit of life is cell. The neuron is the type of cell that composes the brain. And I say the neuron. There are actually many, many, many different kinds of neurons. Neurons specialize for different tasks. Some are just specialized at sending a signal on as far as it can go to the next neuron, and others are shorter or have sort of lots and lots of connections to lots of different ones. So they form sort of network hubs and that sort of thing. And there are a bunch of other types as well. But the basics, every neuron has the same basic structure, okay? Their primary function is to send and receive electrochemical signals, okay? And they do this by, they have a cell body here, which is the sort of main bit, and you can see that slightly darker red circle in the middle of the cell body. That is the nucleus, okay? They have dendrites surrounding the cell body. These receive impulses from other neurons. And then they have an axon, which then branches off, which goes to the dendrites of other neurons. And so a signal comes in this way, gets sent along through the axon to the dendrites of other neurons, which then pass it on to the next set of neurons, etc., etc. The whole system is really interesting. The axon has this fatty covering on it that is called the myelin sheath. And it basically acts like insulation on electric wiring. It helps keep the signal sort of running smoothly down the path. It basically just makes the electrochemical signals more efficient. So they come out at the ends of the axon here at what we call the synapses, which is the space between the dendrite and the axon here. I'm going to point out in a moment that there's some very sort of significant myths about the brain, and one of them is about how much of our brain we use. This is an image of the auditory cortex of a mouse, especially genetically engineered mouse. It's had a chemical sort of added to the brain tissue that turns green, it glows green in the presence of calcium. Neurons send electrochemical signals using calcium channels that we're not going to get into in this class. And this is what a brain working looks like, and you can see there's a lot going on. So anyways, the neurons form up into a network, a massive, massive network of connections between each neuron, right? And one of the great sort of scientific projects that is going on right now is the human connectome project. It's a bunch of different projects, actually, but the goal is to try and map out all of the major connections in the human brain. Um, that's huge. That's huge. There are 100 billion, with a B, neurons in your head. And each of those neurons is connected to many other neurons to form this dense web. That is no small order. Trying to map a network that size on a microscopic scale, that is hard to see the connections unless you've also got living tissue to work with. It's quite a huge endeavor. But some of the hopes of this is that if we can better understand it, we can better understand things like how to heal brain injuries, for instance, how to create prostheses to accommodate people with injury to certain parts of their brain, that sort of thing. It's very exciting. Um, the the groundwork for a lot of this research started with the connectome of the nematode. And we've done this, and it was highly successful. The nematode is a common laboratory organism. It's basically a microscopic roundworm that it's found all over the world, and mostly in soils, but really all over the place. But it has the advantage of only having 302 neurons in its brain. So it's a very, very simple nervous system to work with. Mapping that out required lots of computers to actually process all the possible connections and to see what they all do and how they connect to one another. Now, on the surface, different brains in different animals look very different, but they all share these same features that I've already covered, right? You've got a fish brain on the bottom here, and then 4 different mammal brains on top. The squirrel brain, capybara, lion, and finally a chimpanzee. Chimpanzee looks strikingly like a human brain. You will notice it's smaller. The frontal cortex is much smaller. But there's a lot of resemblance there, right? There's a reason for that, and that is because we all have a shared evolutionary history. Brains build off of the previous brains, so as brains evolve, they're building off the features that were in the previous brains, and the most highly successful structures so far have been these ones, where you've got a cerebellum, a cerebrum, a brainstem, and in this case you can see in the medulla there, as well as having special centers for various functions, like in a fish, smell, olfactory sensation is very important, and so having a special olfactory bulb is really useful, whereas for us as primates, vision is more important. So we have these very big visual cortices, but we have tiny, tiny olfactory bulbs in our brains. Small enough I can't even show you on some of those models I just presented. So, if you get a chance, I will link to this movie in the videos on Canvas. But if you get a chance, watch episode 3 of Your Inner Fish from about the 45-minute mark to about the 53-minute mark, roughly. They cover the early evolution of brains here, and how we go from being these sort of these things that don't have nervous systems and are just kind of like collections of cells, to things that have that can coordinate. The cells communicate very efficiently with one another. It is the origin of your brain and your spinal cord. That's what this pink line here is in the middle, and at the very end is this tiny microscopic bulge, which is the sort of first brain. Okay, I promised some myths. One of the most neuromyths, there are a number of them. I'm just going to cover 2 that are kind of the most popular. There's the 10% myth. Okay, This is the idea that you probably have heard people say, oh, we only use 10% of our brain's capacity, and if we could just use all of our brain's capacity, the amazing things we could sort of do. Not true. We use 100% of our brains all the time. In fact, when you're asleep, you're using your brain more than when you're awake. So, but it's firing what it needs to all the time. There's no wasted part of your brain, okay? It's not like there's all this sort of free-floating sort of processing power in there. All those networks are highly specialized for what they do. So it's not like you can just borrow more processing time from one area or another. This originates actually last century with some psychologists at Harvard who had some sort of dodgy research sort of that pointed towards having some untapped potential and so forth. Has since been the scientific research doesn't support it at all anymore. But the problem, it went largely forgotten until in the 1930s and then the 1960s, later in the 1960s, it was mentioned in a number of self-help books, like Dale Carnegie's How to Make Friends and Influence People, which was a bestseller for years and years and years, and is largely based on a disproven theory about the power of positivity. That's another myth I would love to, we don't have time to bust. So yeah, there's really no research under this. It has no scientific meaning whatsoever. You are using all of the brain you need to use at any time. That's it. Your brain uses what it needs. Otherwise it wouldn't work. The other myth is and this is going to be very near and dear to a lot of people, so I apologize if this one's hard to hear, but the left brain, right brain myth. It's true. What is true is that there is lateralization. There are some functions that are more prevalent on one side than the other. Language is a good one. So a lot of language processing is on the left side of your brain, but actually a little bit is on the right side of your brain too. We know that as I just mentioned, you've got a mental map, both of your senses and your motor functions on each half of the brain, right? The myth is that there are left brain thinkers and right brain thinkers. And left brain thinkers, because there's the language center on the left side of the brain, are more analytical and more verbal and more sort of logical thinkers, that sort of thing. Whereas right brain thinkers are more creatives, and they are more free-spirited and they think outside the box, etc., etc. There is no correlation between the lateralization of any brain function and thinking styles, because we're talking about personality traits here, is what we're talking about. And there's no link between the hemispheres of the brain and particular personality traits whatsoever. As I said, there are neural networks that stretch across both sides for most functions. And that's because, again, evolution uses what it has on hand when it develops. So when a brain evolves new functions, it's just either piggybacking or migrating the function from somewhere else. All of us have the capacity to be both logical and analytical and creative, out-of-the-box thinkers. And you can learn those skills, too. They are skills, not personality traits. The origin seems to come from, again, sort of in the 19th century, there was a psychologist who proposed that the brain is actually two separate entities that most of the time cooperate, but sometimes are in disagreement. And mental illness is the result of the brains being essentially at war with one another. So you've got these two brains that are at war. They have different desires and so forth. In the 20th century, with by the 1960s, we had technologies where we could actually really look at sort of brain function and started realizing that there were things like language were lateralized. It This kind of got combined with this notion that there are two brains, and out comes this left brain right brain myth. So I mean, the bad news is, if you thought you were just a right brain person and that's how you are, you're not. Same with left brain, you're not. The good news is you can learn those skills. All right. We don't know to what degree people have different capacities for that or not. We don't know how to measure that, frankly. But the good news is brains are highly plastic. They change. You can learn things. You can learn new skills and do new things with them. So that's it for this section. Join me next time when we're going to talk about something called encephalization quotient. This is a measure of how big brains are in relation to body size, and it's very important when we're looking at the evolution of brain and brain functions. So until then, take good care of yourself. Have a great week, and I will see you soon.